Yeah, hi everyone. This is Wang. He started well less than a year ago, I guess. So he's in his first year of a postdoc in our group. His PhD, he's from PKU originally, and he got his PhD from University of Washington in applied math. And then he did another postdoc in Paris at the Institute of High Scientific Studies, I guess. they're sort of Institute of Advanced Studies, if you will. And he started here, and he's got a very broad range of interests. This is one area. He specializes in probability theory and mathematical biology. So he's very broad and can work on many different things. So he's going to tell us about SELEX today. Thanks.
Thank you, Tom. So my talk is stochastic model and optimization of SELEX. I will first introduce SELEX, which is a process to select Uptimers. And I will review the traditional deterministic model. And then I build a stochastic model for SELEX and analyze its properties. And last, I will search for the optimal protocol of SELEX. First, Uptimers are oligonucleotide or peptide molecules that bind to a specific target molecule. Some Uptimers have high affinity to a pathogen. Then if we can isolate such Uptimers, they can be used as an alternative of antibodies. In general, we start with a very large library of randomly generated Uptimers, and they have different affinities to the target. And the goal is to select such Uptimers with high affinities in an easy way. So we have a method called systematic evolution of ligands by exponential enrichment, SELEX. We know that Uptimers with higher affinities to the targets are more likely to bind to the targets. This means we can use the targets to pick out such best Uptimers. This protocol is similar to a population evolution process. So this is the protocol of SELEX. We start with some target molecules, the red ones, and large Uptimer library. Here we have different types of Uptimers. We mix them until reaching equilibrium. So some Uptimers are bound to the targets. Here, the blue ones have higher affinities than the green ones. So we have more the blue Uptimers bound to the targets. After that, we remove those unbound Uptimers. And for those targets and some bound Uptimers, we separate them to get those formerly bound Uptimers. And we can see that the proportion of blue ones is higher than the initial proportion. And the last step of this round of SELEX is to amplify such Uptimers with PCR. And we can repeat this procedure to further increase the proportion of the Uptimers with higher affinities. When we start one round of SELEX, we can control the quantity of targets, the quantity of Uptimers, but we cannot change the proportions of different Uptimer types. So like here, we can add more of fewer targets, but for Uptimers, we can only start like 3 and 3 or 10 and 10, but not other proportions, because we need this whole protocol to change the proportion of this best Uptimer. And the goal here is to maximize the proportion of the best Uptimer. I mean, the one with the highest affinity after this round of SELEX. So we need to maximize the proportion of blue ones here.
Can I ask a quick question? So on this background, so this is selection of Uptimers and of course amplification. But I thought at least some protocols of SELEX also involve error-prone PCR to generate additional variants, making it a more true evolutionary process. So when you say amplify Uptimers by PCR, sometimes that involves introducing additional variants.
Yes. For this project, we don't consider the randomness in PCR. So here we just assume that the proportion is the same before and after PCR.
Okay, sounds good.
So for SELEX, a bad choice is to add too many targets. In this case, almost all Uptimers are bound. This means that before and after SELEX, no selection is made, and the proportion of the best Uptimer is invariant. So to find the optimal policy, we first need a mathematical model to study SELEX. A traditional approach uses the law of mass action. This law is valid when the number of molecules is sufficiently large. In this model, big S is the total concentration of targets. Big AI is the total concentration of Uptimer type I. And small ai is the concentration of Uptimers that are bound to targets. K plus and K minus are reaction constants. At stationary, for each reaction of binding, we have this equation. The first term here is the concentration of unbound target unbound target, which corresponds to this term. The second term here is the concentration of unbound Uptimers of type I. So it's this term. And here, this one is the concentration of bound Uptimers type I. So it's here. And if we have different Uptimer types, then we have equations. So given big S and big A, we can solve all these small AI. Now we define K to be K plus over K minus, and we assume K1 is the largest. So this means A1 is the best uptimer. And the goal here is to maximize the proportion of bound A1, which is small a1 over the sum of small ai. And what we can control is big S and big AI, but we cannot change the ratio of A1 over AI. In this deterministic model, we can prove that this proportion increases with the uptimer concentration, and it decreases with the target concentration. Therefore, the optimum policy in the deterministic model is to add as many uptimers as possible and as few targets as possible. So like here, if we have too many targets, almost all the uptimers are bound. So the final proportion of A1 is the same as the initial proportion. And in fact, this one is the lower bound of this proportion. And when we decrease the number of targets, gradually there are more uptimers with lower affinities that are unbound. And finally, if we have very few targets, then these targets do not compete. And the probability for binding to different uptimers are just determined by their reaction constants. So in this case, the final proportion of A1 is this. And we can see that it is the upper bound for this proportion. The problem is that the optimal policy requires very large uptimer concentration and very small target concentration. And when this target concentration is too small, randomness is inevitable. And the law of mass action doesn't hold. Therefore, we need a stochastic model. And we will show that there's something different in this stochastic model. The notations here are similar. Here, S is the total number of targets. Big AI is the total number of AI type uptimers, and small AI is the number of AI uptimers that are bound. And K bar is K over V, where V is the system volume. So K bar is the reaction coefficient. This stochastic model considers a continuous time Markov process on m-dimensional lattice. So each state is the number of bound uptimers, small a1 to am. For this Markov process, the transition rates between neighboring states satisfy this equation. So this one is the number of unbound targets. This is the number of unbound uptimers, and here is the number of bound uptimers. So you can see that this term is very close to this equation here. Given the transition rates, we can calculate the stationary probability distribution, which is this. And for simplicity, in the following, we only consider two Uptimers, A1 and A2. And here, due to stochasticity, those small AI are random. So we need to consider the expectation of this A1 proportion. And we need to exclude the case that no Uptimer is found. So what we consider is a conditional expectation. And this is the counterplot of this proportion. Here, the color is this term, and yellow is higher and blue is lower. The x-axis is the number of initial A1, and y-axis is the target number S. We can see that this proportion decreases with S. However, look at here. This proportion doesn't always increase with the Uptimer number, which is different from the deterministic model. But the same here, that we can prove the expected proportion as the same upper and lower bounds, like the bounds here in the deterministic model. And in practice, if A1 and A2 are zero, meaning that we have no bound Uptimers, since we care about A1, we can stipulate that this proportion is 0. And now we can define this unconditional expectation of this proportion like this. And this is the counterplot of this unconditional expectation. The same x-axis is the Uptimer number and y-axis is the target number. And if the target number and the Uptimer number are both very small, like here, then the probability of no bound Uptimer is too large. So the value of this expectation is small. And here the red curve indicates the optimum target number S for each value of A1. So we can see that for a relatively small A1, the optimum S is not too small. But when we increase A1 to be sufficiently large, the optimum S gradually decreases. And also notice that when we increase A1, the value at this optimum position increases. So it seems that the optimum policy should be making A1 and A2 sufficiently large. And we should make S to be very small. So we could try that A1 to be large and S to be 1. And it's true that this is the optimum policy for one round of SELEX. And the expected proportion reaches the upper bound. The problem is that In this case, we just have one target molecule. This means that after one round of SELEX, we can only get one aptamer type. This means that when we do further rounds of SELEX, there's nothing to select, and the expected A1 proportion does not further increase. So this is the counterplot for this expected proportion after 2 rounds of SELEX. The x-axis is the aptamer number for the first round, and y-axis is the target number for the first round. Here, you can see that if we use the optimum policy for one round of SELEX, which is very large A1 and very small S, like here, then the performance is not quite good. Here you can see that the optimal value of S first decreases with A1, then increases. So we can prove that the optimum policy for multiple rounds of SELEX in the stochastic model is to set A1, A2 to be much larger than S, but S should be also sufficiently large. Then after n rounds of SELEX, this policy has reached this proportion, and we can prove this is the upper bound. And we can see that this proportion converges to 1. And the difference here converges to 0 exponentially fast. And the decay rate is K2 bar over K1 bar. This factor is the most important one for determining the efficiency of multi-round SELEX. So in sum, we first discuss SELEX, which is a process to select the best aptamer for binding a target. In the traditional deterministic model, the optimum policy for any rounds of SELEX is to have a very large aptamer number and a very small target number. And then we develop a stochastic model in which the optimum policy for multiple rounds of SELEX is to have a very large aptamer number, but a moderate target number. This theoretical analysis can be applied to other scenarios, like selecting drug-resistant cells. So that's all. Thank you.